Hello, everyone. My name is Dr. Casey Hewitt, and for the summer, Annie Reyes and I will be your moderators. We are pleased that you could join us this week for our lecture for our first 7-week neuroanatomy mini-series that brings you lectures from experts in the field covering different neuroanatomy topics each week. This series was created by trainees and early career neuropsychologists to provide free, high-quality didactic opportunities. We would also like to recognize our No Neuroanatomy planning group for their hard work pulling this mini-series off. We would also like to thank our sponsors for their financial support for the series. Now, before we start, here are some disclaimers for the series. This is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Questions can be submitted via the Q&A box on the lower left side of your screen, and a recording of today's lecture will be provided on our website later this week. Questions will be asked at the end and not during the lecture. Today's Q&A is led by Annie Reyes. No Neuropsychology is excited to announce a call for members. Applicants who have little to no formal leadership experience and are from historically marginalized and excluded backgrounds in psychology are encouraged to apply. All psychology graduate students, interns, and postdoctoral fellows and neuropsychologists are eligible to apply. The deadline for this application is this Friday, August 13th at 11:59 Eastern Standard Time. Now it is my pleasure to introduce Dr. Robert Bilder for today's lecture titled The Limbic Lobe, a Riddle Wrapped in Mystery Inside an Enigma and Its Closest Friends. Dr. Bilder is a Tenenbaum Family Distinguished Professor of Psychiatry and Psychology, Chief of Psychology at UCLA Health in the David Geffen School of Medicine and Resnick Neuropsychiatric Hospital, and Director of the Center for the Biology of Creativity in the Jane and Terry Simmel Institute for Neuroscience and Human Behavior. He is a board-certified clinical neuropsychologist and directs the UCLA Postdoctoral Fellowship Program in Neuropsychology. His research focuses on brain and behavior and aims to eliminate boundaries between mental health and illness and between everyday and exceptional creativity. His current grants are examining the links of reward system functions in the brain to the genetics of mental illness under the NIMH Research Domains Criteria Initiative and has established a national neuropsychology network to gather big data about the brain. He recently completed the Big C project on exceptional creativity, and he now directs the National Endowment for the Arts Research Lab to measure impact of the arts on well-being, and the limbic lobe has long been his favorite.
Thank you so much, Casey, for the kind introduction. And I'm delighted to be able to come talk to you all today about the limbic lobe. Definitely my favorite of all the lobes. I wanted to share with people that some of you may know our colleague, William Barr. He and I used to work very closely together, and every International Neuropsychological Society annual meeting, we would vote on what was the lobe of the year. I think the limbic lobe is always a big winner and certainly my favorite. In thinking about it, I think one of the greatest things about the limbic lobe is that it remains largely unknown in exactly how it functions relative to some other bits of the brain. And that's why it's been referred to as a riddle wrapped in mystery inside an enigma. When I searched to try to find out who first said that, it turns out that it seems like it was Winston Churchillism who described, forecasting the actions of Russia, which is a riddle wrapped in a mystery inside an enigma. But in looking to try to understand, I'd always thought it was Carl Pribram who used it to describe the functioning of the hippocampus, but it turns out that others have used it to describe the posterior perforated substance, schizophrenia, myoclonic epilepsy. So there may be many mysteries in clinical neuropsychology that are riddles wrapped in enigma, but it'll be fun to talk about the limbic lobe. What do we know about the limbic system? And to start that, I'd like to talk a little bit about the history. It seems that we can see Broca as being the person who first introduced the term limbic lobe into our literature, le grand lobe limbique, basically referring to these tissues that describe the curved rim of the cortex, including the cingulate and the parahippocampal gyri. That brings us back to, the 1850s roughly. But I think that we got more excited about the limbic lobe and its role in emotion when James Pepys in 1937 published a paper called A Proposed Mechanism of Emotion, the anatomical model that was then, became famous as Pepys circuit. And just to, review for a moment, you can see depicted in the upper right there, the classic Pepys circuit, which includes the hippocampal formation, which via the fornix sends projections to the mammillary bodies, thereby to the anterior nucleus of the thalamus, then back to the cingulate, whereby cortical projections then return to the hippocampal formation. And this fundamental functioning of the hippocampus and associated structures, including the cingulate gyrus, were thought to underlie a reverberating circuit that helped to amplify various kinds of processes, and particularly emotional processes, so that Pepys, saw emotional processing as being the core kind of function of this circuit that involves multiple limbic structures. If we look to Wikipedia, we can find that the term limbic system was first used by Paul MacLean back in 1952. And there he described a series of structures that surrounded the cerebral, the boundary between the neocortical hemispheres and the brainstem. And that conceptualization was really made famous in his model of the triune brain. Now, the triune brain concept has a lot of critics in terms of the details of its operation, but I think that there still remains a general consensus that evolution of the brain has proceeded through a series of hierarchical expansions, and that we can identify relatively core brain circuits, which MacLean referred to as the reptilian brain, and that from that core set of brain structures emerged a paleo-mammalian brain, which MacLean referred to as the limbic system, and that this was at the interface between the reptilian brain and the entire set of neocortical developments that ended up being represented in mammals and particularly in primates as being particularly expanded. Now, one of the things that was emphasized, back by MacLean is that some structures, particularly the basal ganglia structures, tend to be relatively conserved across different species, but you can see that the relative dominance in size of these structures tends to change across evolutionary phylogenetic development. So you can see that in the lizard, similarly in the rat and pigeon, in fact, we have pretty extensive basal ganglia structures, and we see quite a bit of development of other limbic and cortical regions in monkey, and that progression probably appears to be even greater in man. It's interesting, though, that despite our sort of corticocentrism in neuropsychology, where we think of humans as having the greatest elaborations of cortical development, that's probably the correct answer, by the way, on board exams. But it's interesting that recent literature has highlighted that, in fact, if we look at the hippocampus, amygdala, and orbitofrontal cortex, all key structures within the limbic system, that these have actually shown quite dramatic expansion in humans relative to other hominid or hominoids, I should say. So if we try to determine how much brain tissue you should have in different areas just based on your size and hemisphere volume, what we see is that the limbic structures are actually quite heavily expanded, and that in humans, and that brings up an important point, which is, well, to what extent have the developments within this limbic system, these structures, been critical to the special functions that humans enjoy, special kinds of functions that the limbic system endows us with. And that's what I'd really like to talk about, is what is the overall structure and function of this wild set of structures that we refer to as the limbic system. I think this figure on the right also highlights what is a somewhat expanded view of what structures are included in the limbic system, including certain aspects of neocortex, such as the ventromedial frontal and the orbitofrontal cortex, along with temporal polar cortex, piriform and entorhinal cortex. And those complement the classic Pape circuit structures of the cingulate, anterior nucleus of the thalamus, mammillary bodies, and hippocampal formation. Now, one thing I think, just to orient us also, is that many of you are probably familiar with the Alexander, DeLong, and Strick model of corticostriatal palatothalamic loops that help us to understand the organization of the cortex as it interacts with all kinds of limbic and subcortical structures. And indeed, it's quite neatly organized. I think that Alexander, DeLong, and Strick originally introduced 5 different loops as if these were discrete loops, but I think they already recognized, and since their time and since the publication of the original paper, we now know that there's really a graded representation that while they identified motor, sensory, associative, and limbic loops, these are not really segregated, but rather they blend into each other, as is depicted, I think, sort of nicely in this particular diagram. And so we can see the limbic loop as a significant segment of all of the corticostriatal palatothalamic loops, and that helps us, I think, see how the limbic system fits in, and how it has representations at the cortical level, at the striatal level, at the palatal level, and the thalamic level. And of course, there are our favorite, accessory structures, including hippocampus and amygdala, that we can't count out there. Now, in thinking about the functions of the limbic system, there's a nice old article from Raj Mohan and Mohandas that gave a high altitude summary. And so they listed a number of the different kinds of functions that have long been attributed to the limbic system. That includes olfaction, sleep and dreams, and then of course the 4 Fs, feeding. Under emotional responses, I think we'd have fighting and fleeing, and sexual behavior is the 4th F. And then in addition, addiction, motivation, memory, and social cognition. Rather than attempting to cover all of these different functions, what I'm really going to try to do today is focus a little bit on the some of the 4 Fs, but then really focus on what we think the hippocampus is doing. How does it subserve memory function in the course of doing all the other things that it does? And then really try to unpack the reward systems a little bit more, which critically involve our limbic loops and associated cortical regions. Just to try to put these things together, it's interesting. I ran across a nice article that tries to highlight how thinking has evolved to some extent from the time of MacLean and his triune brain concept to the evolutionary modeling of Panksepp, and how this maps onto a more modern set of distinctions that have been envisioned by the NIH and the NIMH and their Research Domains Criteria Initiative, which dates to about 2010 and continues to evolve. But there you can see, if we think about the progression hierarchically through evolution, from the reptilian through paleomammalian or limbic functioning up through the neomammalian and neocortical functions, Panksepp's conceptualization brings us from states of panic through fear, rage, lust, seeking, up through high level functions like care and play, while the RDoC initiative has identified fundamental mechanisms that are important for arousal and regulation that we could say occupy some of these very low brainstem centers. But then there's the concepts of negative valence, positive valence, and some social processes that really seem to emerge primarily from our paleo-mammalian brain or limbic system. And then there's the high-level cognitive system, of course, where we in neuropsychology spend the majority of our time. But I think that this diagram tends to highlight how we have a hierarchy of functionality that parallels the hierarchy in phylogenetic development of human brain systems. And the limbic system, just as McLean had said, is really right in the middle of that. So one way to try to understand this, and again, I really favor evolutionary theories, and there's one theory that I'm a big fan of, which is not as popular as I think it should be. And so I'll bring it to you and you can take it or leave it. But it comes from a legacy going back to the 1930s and people who did comparative anatomic studies. You can see those cute little tree shrews in the upper right. I put them in primarily because they're so cute. But Dart had looked at reptiles. Abby was the one who went forward into marsupials. And what they suggested, looking across species, is that there was really a duality in the origins of all of the cortex. And then there was a great anatomist, Friedrich Sanides, who in the 1960s studied a range of mammals, including primates. And he actually looked at the detailed cytoarchitectonic or cellular structure and also connectional anatomy in order to try to understand whether this duality held true. And it was his opinion that there really was there were dual trends that were represented in cellular structure and connectivity. And this was subsequently elaborated quite extensively by Deepak Pandya and a number of other colleagues from the 1980s up until the present. And their studies of connectional anatomy and cytoarchitectonics really make it clear that this duality in the organization of all brain regions is really quite profound and really helps to simplify our understanding of the functional anatomy of the entire brain. And here's for those who are spatially gifted and like to see the pictures of these things. I think we've got a nice depiction of this dual trends hypothesis right here. And what you can see is that there are really two trends, one of which originates in the primitive hippocampal formation. And this is called the archicortical trend. It starts in the hippocampal formation and arches up through the cingulate gyrus and then up over the dorsal surfaces, the medial and dorsal surfaces of the brain, where it then comes out over the dorsal surfaces of the cortical regions in both frontal and parietal lobes. In contrast, there's another primitive olfactory trend, a primitive cellular moiety that begins in olfactory cortex and then arches out ventrally and laterally. This is called the paleocortical trend, and it tends to occupy the ventral regions of frontal and posterior cortices. And the integrity of these two trends and the connectivity that honors the formation of these trends has been really beautifully detailed by Pandya and colleagues. And now you may wonder, well, how does this relate to the limbic system? Well, with the limbic system, we've got really two big bundles that relate the limbic system to the relevant cortical regions. We've got the cingulate fasciculus that really preferentially connects limbic structures to the dorsal trend or the archicortical trend. And there you can see, how the cingulate interconnects all these regions. And separately from that, we have for the paleocortical trend, the ventrolateral trend, the uncinate fasciculus that connects these limbic regions to the appropriate cortical components. And here you have, for those again who like to see prettier pictures, we have a nice mapping where we stripped away the cortex so you can see the white matter bundles And there you can see the dorsal cingulate trend and the ventral uncinate fasciculus trend. And this highlights the connectional anatomy that really continues to honor this dual origins of all the cortex by connecting to the relevant limbic structures. So let's dig in a little bit more to how these different sets of structures work together. One point I'll make, and there's a very nice article from 2012 from Gitirian and colleagues. It includes Deepak Pandya and Michael Petrides, who many of you may have may be familiar with. And the key points here is that the dorsal and medial archicortical trends really are connected to the dorsal and medial. The limbic regions and the frontal regions are very closely connected. And in contrast, the lateral and ventral visual association areas, somatosensory areas, auditory association regions, they relate preferentially to the lateral and orbital and then ventromedial prefrontal areas. And the ventral, multimodal, and limbic areas also relate more strongly to these ventrolateral and orbital prefrontal regions. So I think that the overall honoring of the dual trends is something that has characterized not only the cortex, but also these limbic system components. Now, if we think about how does the duality of this anatomic organization relate to dualities in functional organization of the brain, I'd like to bring you back to the 1960s when Arya Rautenberg identified that there were really two different kinds of fundamental arousal systems in the brain. As you may recall, back in the days of Maruzzi and Magoon, let's go UCLA, go Bruins. But anyhow, Maruzzi and Magoon identified the reticular activating system, series of brainstem structures and network that, if stimulated, could excite the brain and wake up the animal, and if silenced, could also quiet the brain, decrease overall arousal, and the animals would go to sleep or go into a coma. But what they discovered is that, what Rautenberg discovered is that there was really not just a singular arousal system. The RAS was just an ascending phasic system. In other words, you would stimulate that and it would lead the animal to wake up very briefly, but then they would go back to sleep as soon as they were not stimulated anymore. It was a brief, fleeting, or phasic kind of activation. In contrast, what they found is that there was another set of structures really centering in the basal ganglia that underlie tonic activation or a steady activation. If you stimulate that, the animal would stay awake and conscious for an extended period of time. And this fundamental distinction between phasic and tonic arousal is something that characterized forebrain versus ascending kinds of activations. And Karl Pribram, the late Karl Pribram, bless him, had related this to dimensions of cognitive control, and one that he referred to as the stabile versus labile dimension, which I think is critically important in all of neuropsychology, to try to understand the brain systems that are involved in stabilizing cortical activation states versus those that are involved in creating novel activation states and detecting novelty in the environment. And so Pribram described the bias of different systems in the brain as being redundancy biased or novelty biased, where the redundancy biased ones were the ones involved in tonic activation. Those are the forebrain controls. Those are the controls that are involved in the projectional control of behavior and the formation and stabilization of behavioral programs despite interference. Whereas there are novelty bias systems that are really critical for detecting novelty in the environment, looking for changes in the stimulus world that may be important for us in particular context in order to change our activation states. For those who are interested in this theory, we have an article from 2012 that highlights how this fundamental functional distinction between stability and flexibility maps onto the archicortical versus paleocortical trends respectively, so that you basically have the dorsal and medial archicortical system that is for stability, and the ventrolateral paleocortical system for flexibility. And this just highlights some of the distinctions that we identified between the paleocortical, that's the ventrolateral, and the archicortical, that's the dorsal medial trend. Again, for those who are interested, you can see how this really is a major simplifying assumption that helps to understand lots of brain activation processes. In my opinion, this is critically important in trying to understand how these different components of the limbic system function together in the detection of novelty and the stabilization of behavior. And that's what I'd like to dig into a little bit. Again, for those who want to know a little bit more about how this may relate to certain other biological principles, you could check out this other article we wrote that relates tonic and phasic dopamine transmission to exactly the same set of tonic and phasic arousal processes. But to further elaborate, how I think the limbic system helps to mediate the engagement of either stability or flexibility in the brain, I want to resort to a conceptualization that was made popular by a guy named Stephen Grossberg. Stephen Grossberg developed what he called adaptive resonance theory, and I think this is a nice way to describe how the brain works quite generally, and using sort of computational modeling. Basically, what we have is, if you look at the F2 system, that system is really in charge of what you might call behavioral programs, that top-down functioning that sets our expectations. Well, there's a bottom-up system that comes in through your sense organs, and that basically comes from our posterior cortex. And there's an extensive cross-wiring between the expectation systems from F2 and the incoming input systems from F1, which represents reality. And the resonance of these two systems indicates that all is well and that what's coming in through the input systems matches reality. But if what's coming in through the F1 system, through the inputs from our sensory systems, doesn't match our expectations, then there is the generation of a reset wave, and that leads us to change our expectations and increase the potential for novel behavioral programs, and shake up basically the entire brain until a new activation state is arrived at that helps to minimize the mismatch between the expectations and the perceptual world. And if we look at that attentional subsystem, we can see that as basically being the frontal and posterior cortices, the orienting subsystem following Rautenberg. I would say that zone A is the hippocampal formation. And one of the things the limbic system does is serve as a conduit for detecting cortical mismatch, sensing novelty in the environment and how it differs from our expectations, and then either stopping our ongoing behavioral programs or stabilizing those behavioral programs that maximize the matching of our expectations. Now, a lot of this work on resonance and stabilization can be done just at a corticocortical level between the F1 and F2 systems. There is very good evidence about the anatomy that connects the frontal and posterior cortices together. There's feed forward and feedback connections, and that enables very rapid correction of minor mismatches. That's sort of automatic error correction. For example, the movement of our eyes, just to keep track and stabilize images in our visual cortex. But what if something really unexpected happens? If you run into a snake, or a lion comes out of the woods, or if your grant application is not reviewed favorably, then what that does is it leads to a mismatch process that causes a reset. This is when the hippocampus goes into a theta rhythm, and it's in that theta rhythm, that detection of mismatch, which reflects the posterior cortex not matching what's expected as predicted by the frontal cortex, that we see the hippocampus going into theta and causing a change and an introduction of overall flexibility in cortical activation states, again, until a new state can be achieved that minimizes that kind of failure. Now, this is, may seem to have come from, very arcane and ancient hypotheses. And novelty detection. Now, I'm not going to spend a lot of time talking about emotional responses, but one of the things I will say about that is that when the hippocampal formation is detecting novelty in the environment, if the in mammals is what's called the orienting response. We see that our sensory thresholds become lower, our pupils dilate to let in more light. We get a little bit of a piloerection of the hair on our arms, and our body will stand up a little bit. Our auditory thresholds actually reduce a little bit so we can hear quiet sounds better. Heart rate decelerates. All of these functions go along with the hippocampal theta and its response during the orienting response. Now, if the mismatch is greater, though, and that is when the amygdala is engaged, and we typically will go beyond the orienting response to engage other kinds of responses, including defensive responses. And so that's where this high level of mismatch is involved in generating fight and flight responses. I see the IT support services raised their hand. Are we having a problem with the IT?
No problem. I think someone just may have joined in with that name. Yes.
All right. All right. I was afraid there might be a major mismatch that was resulting in amygdaloid activation, but I guess not. So, so I think that what that highlights is a distinction where low levels of mismatch generate interest in and increased sensitivity to the external world, whereas if the degree of mismatch exceeds some threshold, that's when increases in sensitivity will go away. What we see then is pupillary constriction. We see heart rate acceleration for action, to facilitate action. And that's when these very much more brittle responses that involve fighting or fleeing are engaged. But anyhow, back to the hippocampus. What's really interesting, and people had a hard time understanding, is how could the hippocampus possibly serve both in detection of mismatch and in the representation of familiarity. And what I think this new information is revealing is that if we look at the electrical potentials and the cellular structures that are involved in generating those potentials, what we can see is that both detection of mismatch, and there the P300 is the sort of electrophysiological sine qua non of mismatch detection, where we have an oddball stimulus. The auditory oddball is easier to say online. So if we have beep, 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 boop, beep, 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 we tend to orient and habituate to all of the beeps, but when the boop comes on, it generates a mismatch detection process that is maximal around 300 milliseconds. In contrast, there's also a hippocampal positive 600 millisecond potential that tends to respond when we recognize a familiar stimuli, for example, famous faces. And Barbeau's hypothesis is basically that you could see this actually across a whole bunch of different oddball tasks and different memory tasks, where the memory tasks tend to be a little bit delayed relative to the oddball tasks, but you can actually identify, the differences between oddballs and famous faces based primarily on the timing and to some extent locations within the hippocampal formation. And Barbeau's hypothesis is that this distinction is really because of the distinction between a monosynaptic pathway in the hippocampus, where the temporomonic projections that come from entorhinal layer 3 get to the pyramidal cells of the CA1 field, the corno-amanus field 1 at the apical level. And that creates this negative current sink. And the idea is that that's really like the current state of the world. And in contrast, there's this trisynaptic projection from entorhinal layer 2 that reaches the pyramidal cells of corno-amanus field 1 at the basal level. And that creates a positive current sink or negative current source. And it's that circuit that's thought to be associated with the autoassociative processes that are linked to memory retrieval. So the idea is that we've got both of these kinds of potentials, but they occur at a different phase of hippocampal excitation. And probably there are different phases of the theta wave that are responsible for both mismatch detection and familiarity detection. It's just a matter of where and via which circuit the activation is occurring. One thing that is important to recognize is that it's unlikely we're going to see how these processes unfold using things like functional MRI, because they're both going to cause increases in bold response. I think that what we're going to need is more electrophysiological studies, detailed, depth recordings while people are undergoing temporal lobe surgery. I think the idea overall is that these hippocampal theta rhythms probably are responsible for both encoding and retrieval processes. It's just they occur at different phases of the theta rhythm, and that those don't overlap. And there's a nice model that's separate phases of encoding and retrieval model by Haselmo and Stern that you could check out about that. So I think that trying to summarize this briefly, the fundamental idea is that the hippocampus participates in the transition between these two neocortical large-scale mutually exclusive networks. One is involved in memory, the other in target detection. And the reason for their exclusivity is that oddball tasks require attention to external stimuli, while memory tasks require attention to internal representations. And interestingly enough, this is, I believe, a reimagining of a hypothesis originally made by Pribram and McGinnis back in 1975, and that is that the hippocampus involves an uncoupling of arousal and activation, and that it is actually the hippocampus that determines whether we're going to stabilize or labelize our current pattern of network activations. So the hippocampus serves like an arbitrator of whether the current situation calls for stability or flexibility, largely based on detection of mismatch and familiarity processes. So here's Carl saying the hippocampal circuit uncouples the stimulus from the response by coordinating the amygdala and basal ganglia mechanisms so that appropriate changes in the central representation can occur. And he described this as a process that entails effort, because changing the state of the cortex is actually work in a physics sense. And so he saw really the amygdala as being the primary motivator of change, and the basal ganglia the primary driver of stability in these systems. Now, just to highlight that there are many other theories of hippocampal function. In my view, there's a lot more overlap among these different theories than there are distinctions, but we have Rolle's theory, which he's elaborated into a computational model. I think that these models all overlap in suggesting that what we've got is a big funnel of posterior cortical signals coming in through the entorhinal cortex, through the dentate gyrus, into CA3, then to CA1, and then via subiculum back to modify the cortex. I think that this, is not so much different from the kind of classic reverberating circuit that's been envisioned as a core function of limbic functioning for many years. But I think we've learned a lot more about the unique roles of CA3, CA1, and the internal hippocampal circuitry. I won't go over more of these details because we want to get to talk about arousal, but I'm going to have the slides available for everybody so you can check out a number of these other theories as well. Localization of the hippocampal engram, so to speak, but this helps to reconcile memory indexing and cognitive map theories, all of which I believe are compatible with what we've talked about so far. But basically, the idea is, from the CA1, we have a reverberating circuit activation of the cortical representations of experience. That's how the hippocampus plays its role in supporting memory retrieval. There's also a dialogue about whether parts of the dentate gyrus on their way to getting into the hippocampal function are involved more in pattern separation or binding of stimuli. And I think that, it's probably, both. You could probably, I think it's more a matter of perspective. This particular group of authors believes binding better captures the contribution than pattern separation, but I think they're really converging on largely the same things. And finally, yet another theory, the CRISP theory, really focuses on pattern completion, context reset. And again, I think all of these theories really focus on the same kind of overall patterning of cortical inputs at different levels into hippocampal formation, whether it's via the dentate gyrus and CA3 to CA1, or via this more direct pathway into CA1 that ends up feeding back via the entorhinal cortex to the rest of the cortical fields, and helping us to both detect and then sustain patterns of activation in large mapped regions of the corresponding cortical territories. There's a sublayer specific perspective that I think is very helpful, along with having these beautiful pictures for folks who would like to get into the detailed anatomy of this, and also to highlight how different phases of the theta rhythm, as I was suggesting before, probably map onto different patterns of activity within the hippocampal formation. And I think this paper really highlights how that works, exactly which regions are involved at which phase of theta rhythm, and what are the different components of anatomy that are probably involved in that. So this is, I'm going to guess, more than anyone wanted to know about how the hippocampus works, but my big hope would be that you got an idea of the overall complexity of what the hippocampus is probably really doing, which goes pretty far beyond, I think, the kind of, the old view that the hippocampus is just involved in episodic memory function, which is, way more simplification than we need. And what I'm bringing to you today is probably more complication than most of you will need as neuropsychologists, but I hope that those sources will help you to track down what the hippocampus is doing to whatever depth you want. Now let's switch to talking about reward systems. Whoops, let me come back to this. One thing I wanted to highlight about reward systems is that, again, a lot of people think that, oh, we're just talking about like the dopamine signaling from the ventral tegmental area up to the nucleus accumbens, but the reality of it is that there's a lot more than dopamine involved here. We do have those VTA to nucleus accumbens projections, but there's also a serotonergic pattern that's really important in reward. We have GABAergic pathways that are critical, peptidergic pathways, glutamatergic pathways that are critical. So I like to show this slide just to highlight that we've got quite a lot more going on than just this dopaminergic projection. But let's focus on that dopaminergic projection for a minute, because I think it's really critically important in understanding the limbic system and its interaction with the striatum and cortex in trying to mediate reward. And this is central to the research domains criteria both negative and positive valence systems. So I think that, I want to try to highlight a little bit of that. A core element of that entire theory, again, comes back to detecting mismatch between what we expect and what actually is occurring. So if there's one take-home message from this lecture, it's, gee, comparing what we expect to what is actually happening is a critical function of the entire limbic system and its friends. And it is used for a number of very important purposes. One set of purposes is for memory functioning. The other set of purposes is really for reward functioning and the guiding of behavior in that way. So anyhow, Wolfram Schultz is largely credited with the discovery of what we call prediction errors. And basically, if you're expecting something, there's three possible outcomes. Either the reward comes when it's not predicted, or it's better than you predicted. That's when you get a positive prediction error. If reward comes just when it's predicted, that's the status quo, not much happens. And if reward does not come when you predict it, yuck, that's a negative prediction error. And remarkably, these prediction errors map onto the activity of the ventral tegmental area, which then secretes dopamine. So you get even more dopamine transmitted up to the nucleus accumbens, or less, depending on whether you get a positive or negative prediction error. So it's quite a remarkable and relatively simple hypothesis that turns out to hold up pretty well. This is basically the the prediction error hypothesis in a nutshell, as shown by Wolfram Schultz. And he's done a bunch of really elegant experiments showing not only do you get these prediction errors, but you can show the transfer from one stimulus to an earlier stimulus. There's a ton of really cool experimental studies that have been done, and including this modeling of this kind can enable us to tell the difference between an organism's response to safe choices or risky choices, and the circumstances under which we're likely to pick safe versus risky choices in our behavior. And so this is very much related to a whole bunch of theories of impulsivity, gambling behavior, addictive behavior, and so on, including quite importantly, the prediction of delayed discounting behavior, where we tend not to take as seriously those kinds of things that are going to happen a long time in the future, or where we think it's a low probability. That's the only reason why we can, how we can explain why people continue to smoke cigarettes, for example, after they know it's certainly not the healthy thing to do. But you're going to get cancer. Well, it's not 100%. Maybe it's a low probability you're going to get cancer. And then it's also, I'm not going to get cancer now. I'm going to get cancer maybe in 20 or 30 years. So those mechanisms are really nicely explained by these hypotheses. And given the time, I'm going to try to keep on going forward and just highlight a couple of the articles that I think can really help you to unpack some of the limbic system and associated cortical contributions to reward-guided learning and decision-making. This paper is one that I think is really nice and helps to simplify things remarkably, from Rushworth and colleagues. And there they look really primarily at monkey brain functioning, and have isolated just 5 components of circuits that they say are critically important in reward circuitry. I'm not even going to talk about the anterior frontal polar cortex, but I would like to talk a little bit about the ventromedial prefrontal cortex and the medial orbitofrontal cortex. These regions are probably more associated with the value of rewards. This is very much related to the concept of positive and negative prediction error. And probably both positive and negative values are being represented, and its decreases in signal represent loss, while increases represent gain. As I was mentioning before, a decrease in signal also associated if there's a delay before the reward is given, and that helps to explain delayed discounting. And here, if you didn't believe me, here's a nice, colorful image of the brain showing that this kind of hypothesis can be well mapped, even in human brains, based on the pattern of response versus when you get the outcome, and whether the outcome is positive or negative. Now, interestingly, the lateral parts of the orbitofrontal cortex seem to be more activated by punishment or non-reward, and seem to be involved in credit assignment. So there we have the association of values with specific stimuli. So for example, monkeys is quite interesting. If you train monkeys with peanuts or raisins, they tend to like both. But if you give a monkey a lot of peanuts, they like raisins more. And if you give a monkey a lot of raisins, they like peanuts more. So if you devalue one of them by overfeeding, the lateral orbitofrontal lesion animals don't pick the one that would give them the preferred foods. They lose their ability to discriminate those rewarding stimuli. So it's interesting that this seems to be segregated out to the lateral orbitofrontal cortex. And then the anterior cingulate seems to play a unique role, distinct from these other reward detecting centers. And here it really seems to be more involved in cost benefit decision making. So if there's a conflict, then it's probably going to require engagement of the anterior cingulate cortex, which also will adjudicate whether or not what you need to do to get the reward is worth it. So what's found is that a lot of effort measures, if you have a more effortful approach that may give you a slightly greater reward, that will be evaluated and engage anterior cingulate to try to figure out, is it really worth it, the effort to engage in this behavior? And it probably is coordinating with the value information from the ventromedial prefrontal cortex and medial orbitofrontal cortex of what those delay costs are likely to be. Now, there's, again, a quite fancy anatomy, well linked to understanding how these circuits operate. I think that I highly recommend this article by Haber and Knudsen to you if you're interested in really delving in. Again, just as we were talking previously about the complex corticostriato-palliothalamic loops that are important in understanding all cortical and subcortical behavior. I think that the engagement of amygdala, hippocampus with the ventromedial prefrontal cortex and the substantia nigra, and particularly with the different components of the basal ganglia that are related to those is really well laid out, and we can see that the ventral striatal or nucleus accumbens kinds of activations they're coordinated with the ventral tegmental area to signal reward and to show positive and negative prediction errors is quite neatly mapped onto the combination of hippocampal and amygdaloid functions. One other key point I'd like to make is that it's probably from this shell region where you see the big S at the most ventral part of the ventral striatum, projections there to the hypothalamus and the bed nucleus of the stria terminalis may help to understand the role of these reward and limbic system outputs in the regulation of visceral and autonomic behavior. And here are even prettier views that show, again, this is a more graded kind of a set of representations. We can see there's really a neat progression from the dorsolateral prefrontal through the dorsal anterior cingulate through orbitofrontal to ventromedial prefrontal contributions as we go from the more dorsal parts of the striatum down to the more ventral parts of the striatum. I think I'm going to skip through a few of these other really cool slides that really just show more how all of those regions are integrated with ventral tegmental and substantia nigra regions, leave a little bit of time for questions. So I think that if I talk more about this, it will just be too overwhelming. So thank you very much for listening.
Thank you so much, Dr. Builder. That was a really wonderful presentation. And definitely it will be nice to go back and look at the slides, look at those papers with, going over complex concepts. So we do have a few questions. We're going to keep it short, not to go over the hour. The first one that we have here is, With the many clinical disorders involving the limbic system, what are some recommendations you provide to patients who have a disorder involving the limbic system and are having some cognitive or emotional problems?
Yeah, I'll take one thing that'll give me an opportunity to talk about something that we didn't talk about yet, which is anxiety. So one of the sort of cool things that relates our understanding of hippocampal functioning to psychiatric disorders, and also that relates very much to what I've just been talking about, is what Jeffrey Gray in his neuropsychology of anxiety referred to as the septo-hippocampal stop system. This is basically the same system that Pribram was talking about, where the hippocampal formation, if it detects mismatch, causes us to stop what we're doing. Mm-hmm. And when we stop doing what we're doing, that is the experience of anxiety. When the lion suddenly appears on the jungle path, that is what's going on. So a lot of times I will help patients understand the link between anxiety and their memory problems, because we all know as clinical neuropsychologists, what's the problem? No matter what's wrong with them, they always come in with concerns about memory. Oh, my memory is no good. It's my short-term memory. So we have to try to understand, well, what do they mean by that? But what I find is that it's very helpful to people to understand the interplay of anxiety with memory disruption. When we have people who get derailed and lose track, and become their own worst enemies in the course of memory and coding and retrieval because of anxiety. Mm-hmm. So I explained the septo-hippocampal system to them, and almost every patient can immediately grasp what I'm talking about. Says, oh yeah, that's right. When I get worried about something, I stop doing what I'm doing, and then it disrupts everything. You're not going to learn anything new at that point. And so I found that to be a very helpful way that, what's really a complicated neuroanatomy and functional neuroanatomy can be converted to a pretty straightforward message. So for patients who like to have brain explanations of their behavior, I find that to be really useful. Also had good luck with people with borderline personality disorder explaining orbitofrontal inhibition of amygdaloid activation, because the amygdaloid spiking theory of when we get triggered into fight or flight responses, usually that kind of spiking gets inhibited by orbitofrontal cortical regions. And in fact, that's one of the virtues of the second signaling system, the language system, is to modulate and give us a structure to help to regulate other lower level limbic impulses. And so I will tell people, if they have borderline personality disorders, well, one way we understand this is that there's these set of cortical regions that are usually involved in the regulation of this limbic excitation. And that if those things aren't developed well, sometimes through chaotic early environment or through the loss of attachments that help to support our faith that things are going to turn out well with other members of our species, then we may have these excessive reactions.
Yeah, and I'm sure that's very helpful, with the stigma that's associated with this disorder, is to have that, connection with the actual brain and what's going on. I'm sure patients may find that helpful.
It has worked for me.
Thank you so much. And I guess there's another question related to that. How do you describe the limbic system during feedback sessions in a way that it's more understandable for patients?
Yeah, so it depends entirely on what's wrong. And so I just, those two examples where, anxiety or things like, excessive anger, or expressions of frustration may be understood through modeling these things. With people with memory problems, we'll often talk about, what it is that we think the hippocampus is doing and these structures are doing with memory functioning. And I find it helpful to patients, rather than thinking that memories have been erased, which I think many people believe. And of course, that may be true, that some of the cortical capacity to even represent memories at all will be lost ultimately. But I think especially in early stages of memory dysfunction, where the cause of the dysfunction really is more limbic, we can talk about partial activation. Say, well, your hippocampus isn't available to help to trigger these things the way it normally would be able to. But You can use other things like old pictures, and repetition of material so that it brings things to mind more and helps to make more robust the interconnected patterns that will then be easier to excite and retrieve in the future. So rehearsal, cueing, repetition, these are the kinds of things that can be helpful because of the way that memory system may be dysfunctional.
Thank you. Yeah, that's an excellent recommendation. Well, we have a few more questions, but now that we're at the hour, we just want to make sure we're not taking more of your time. We appreciate you coming and giving this excellent lecture. Our viewers, definitely, we had over 100 viewers today. So thank you so much. And for those who are watching, the lecture as well as some slides will be available on our website.